Hi. So in previous session I have discussed about how to create the knockout mice, and whatever I have discussed, this is for making single gene knockout mice. So this is a very interesting how you will make the, if you want to knock out two genes, how you will go for that. So the answer is not very difficult. First you need to make a single knockout mice for each gene, okay, and after that you can achieve very simply just by crossing, okay. You need not to knock out both gene in one embryonic stem cell. If you will do, most likely that embryonic stem cell will be severely damaged, and you cannot get the embryonic stem cell which has both gene alleles will be disrupted, okay. So the most easy and convenient way is that you can create a single knockout mice for each gene, for example gene 1 and gene 2, and then you can do the mating, and then by that you can get the double knockout mice, okay. It's in a very simpler term, if you, which you have studied very well in maybe class 12th or 10th standard, or maybe in graduation. It's a simple dihybrid cross, okay. So by dihybrid cross you can achieve this this double knockout mice, okay. So I am not going to talk about the Mendelian law and monohybrid, dihybrid or trihybrid cross, okay. So it is also possible to make a trihybrid also. You can make a triple knockout mice, but it takes a huge effort. You need to do a huge screening, okay. It's not generally it's not so much recommended. Generally we use mainly single knockout mice. In general we use single knockout mice, rarely we use the double knockout mice, okay. And if you are interested what is the use of double knockout mice, then maybe during discussion I can explain if you want, okay. Otherwise no need to go for that double knockout mice experiment, okay. Now I will take you further in the transgenesis, and now I will explain you the how to create the conditional knockout mice. What are the prerequisite for making conditional knockout mice? And first, or very simple question, why you make the conditional knockout mice? So I think in previous session I have explained you. So if the gene which you have chose, maybe it is involved in some development process, okay, and if it is involved in development process, then this, and since you have disrupted this gene, then the embryo will not develop, and there will be a death of embryo, in utero death of embryo, okay. So in that scenario, and if you want to study that gene in particular cell type, then you need to make a conditional knockout mice, okay. And how we achieve? We achieve there is some molecular techniques, and I will explain you in this session. So this is I have already introduced about the embryonic stem cell, so no need to explain. Here I am again coming with some example. The same gene here, you can see this is a gene for that IPS or MAVS gene, okay, and these are the 5' UTR, and which is represented in blue color, and the pink color is the exons, okay, and you can see there is a 3' UTR, okay. So here the task is, I believe it's at least 2 times more difficult, and overall if I see, making of conditional knockout mice is at least maybe 3 times or 4 times difficult. Okay, so you will understand soon. Okay. So, here this is a genomic arrangement of the gene IPS, and here we need to make a targeting vector in this fashion. Now, you have you understood about the HSV-TK, you understood about the neomycin, but here you can see there is some more red color boxes. Okay. So, what is this red color boxes? So, this red color boxes are LOXP site. Okay. I will explain you what is LOXP site, but before that you should understand this is a very short stretch of nucleic acid. If I remember, I think it is a 34 base pair. Okay. So, this you need to incorporate this 34 base pair in the genome. Okay. Then you can achieve the conditional knockout mice. Okay. Then you will achieve, I will say, 50% of conditional knockout mice. So, what is LOXP site? I have a one additional slide for LOXP and LOXP sites. So, when you incorporate this LOXP site, then there is some enzyme which we call it as a Cre enzyme. Okay. So, this Cre enzyme will basically chew off in between two LOXP site, chew off the DNA. Okay. So, in that way there will be a deletion of that gene. Okay. So, this deletion will be achieved when the Cre enzyme will come in action. Okay. Now, you might have a simple question, from where this Cre enzyme and what is Cre enzyme? Okay. So, this Cre enzyme, from where it will come? So, I will explain you in a later slide. So, once you make this kind of targeting vector, then you will put it inside the cell, okay, embryonic stem cell, and finally you will achieve this kind of mutant allele for that gene. Here you can see that there are only two LOXP sites. Okay. And it is far more difficult. Why I was saying? Because when you are making the knockout mice and when you are cloning a fragment of DNA, if you remember the previous session, if you don't, please relook that previous session. Okay. Then at that time, when you were cloning this big chunk of DNA in which will facilitate the recombination, that when you are cloning, then it is far more easier. Why? Because when you are cloning that fragment, you don't care about the sequence. Okay, you know that how you clone the gene. You basically do the PCR amplification, and please remember, PCR amplification is error prone method. Okay. So that is even if some mutation is incorporated, it's not bad, it's rather good. Okay. That the gene will be much more disrupted. Okay. Not only the 5' UTR and few exons, but also it will disrupt if there is some mutation in the exons or wherever, then that will disrupt much more efficiently, right. But here you can't afford to incorporate mutations. If you will incorporate mutation, then that will be not good. Here my aim is not to disrupt any sequence, genomic sequence, and in that circumstances you have to clone a big chunk of DNA, say 10 kilobase pair, 11 kilobase pair, or maybe 12 kilobase pair, which is very difficult. If you have any experience for doing experiment, cloning more than 1 kilobase pair is very difficult, and here it is, the size of DNA is huge, right, 10, 11, 12, like that. It's a very difficult, and there is a no I am not very confident that okay, in one shot you will be able to amplify such a big DNA fragment. Even if you amplify such a big DNA fragment, then that will incorporate lot of mutation, because PCR is an error prone method. There is no proofreading. Okay. So this is a far more difficult. Making a targeting vector for conditional knockout mice is itself is a big challenge. And even if you made it, then there is a second level of challenge. So once you incorporate this targeting vector inside the embryonic stem cells, okay, then there will be another level of challenge, that you need to remove this neocassette or neogene gene segment. And how you will achieve? You basically you transfect or you introduce a plasmid which is expressing a Cre enzyme. Okay. So when in this scenario, you just look at the targeting vector. In this scenario, if you put and consider that this HSV-TK is removed, so rest of screening will be same as you have learned for knockout mice. HSV-TK will be removed and this whole chunk of DNA will be in the genome, okay. But there will be a neomycin, okay, and you need to remove this neomycin. And when you will incorporate the Cre enzyme in the cell, you will introduce a plasmid which will express the Cre enzyme, okay. This is a simple way, or this is the only way I will say, okay. So when you will introduce the Cre enzyme, then there will be a several possibility. One possibility is only neomycin cassette will be chewed off. Another possibility is between the 5 prime end of neomycin and the 3 prime end of or in between this 5 prime end of neomycin and 3 prime end of neomycin and 5 prime end of this gene will be chewed off. This is also a possibility. Or another possibility is the whole thing will be chewed off. So all these are the multi-level complication, and it is not very easy to get out from this. Okay, you have to keep on trying, and then you have to have this kind of situation, the mutant allele, okay, where only LOXP sites are there, which is 5 prime, which is upstream to 5 prime of this UTR, okay, and there will be a LOXP site which is downstream to 3 prime UTR of this gene, okay. Once you have this thing, then you have achieved. But please remember again, when you will do all these things, all manipulation and all these complicated experiment, your embryonic stem cell may differentiate, okay, and then you can never achieve the you can never get this kind of mice. You can have the cell, but cell will not become a mice, okay. So these are the different levels of challenges which I am explaining, okay. So once you successfully, of course people got this conditional knockout mice, it's not a new thing, it's a quite old thing, okay. People do made conditional knockout mice, but it is extremely challenging in both financial and labor intensive and all kinds of things, okay. So once you get this kind of mice, you will of course do screening and all those things, and finally you will get the mice with LOXP site which you want to delete, okay. And when you will do this thing, then this mice will survive, okay. During development there is no change in the, everything will goes as per plan of the cell, okay, in a very normal way, okay. Now you want to delete this gene in say particular cell type or in particular organ, then over there you need to make a Cre knock-in mice, which is again another set of creation of transgenic mice, and it is again very difficult. You have to, for example, you want to knock out this gene in lung cells, or for example you want to delete this gene in B cells, okay. So what you have to do, you have to find out the gene which is selectively expressing in that particular cell type. that is for example B cells. Okay. So, there are some molecule which is specifically expressing in that cell. Okay. So, under that promoter you have to put the Cre gene. Okay. So, once you have this Cre mice, okay, for example B cell or macrophage or dendritic cells, whatever, you have to find out the unique gene which is exclusively expressing in that particular cell type. Okay. Once you have that kind of gene, then you have to make you have to introduce the Cre gene, and then you have to create this Cre mice. After creating this Cre mice, you will mate them. Please remember, you have to have both alleles should be mutated. The LOXP site should be present in both allele. I will show you. Let me explain what is this Cre and LOXP thing. Okay. So this is a Cre, and Cre is basically a recombinase which is very small protein, 38 kilodalton, and this is a LOXP site. Okay. It's a 34 base pair. Yeah. And you can see that there is a palindromic sequence, and then there is a intervening sequences. Okay. So basically this site, this kind of sequence you need to incorporate into the genome, and from where this LOX Cre and LOXP system was discovered, it's basically derived from the P1 bacteriophage. Okay. You know bacteriophage, okay, which the viruses which infect the bacteria. Okay. So from there this is derived, and this is a very beautiful system. This is a really very good discovery. Okay. So using this system you can do a lot of genome manipulation very easily. Okay. For example, if you put this LOXP site in this orientation, for example, you can consider this is a orientation 5' and 3' end of LOXP site. Okay. So if you put it in this orientation, then the intervening sequence will be chewed off. Okay. But if you change the orientation, then the DNA fragment will be flipped by Cre enzyme. Okay. So you can do all those manipulations. And if you have this kind of situation, okay, if you put this 4 LOXP site in this orientation, then there will be a translocation of the intervening DNA sequences. So this is a very beautiful system, I will say. Okay. So once you have this LOXP mice for that particular gene, then you can mate it with Cre mice. Okay. There I have a very good slide which can explain how you achieve. So this is a tissue specific Cre. Okay. I have told you the gene which is specifically expressing in particular tissues or cells. So you have to incorporate Cre. Here you can see there is a under that promoter you have the Cre gene, and that will make protein. And this is a floxed allele for gene Y, for example, the gene which I have explained you, maybe IPS1 or MOFs. So you have this kind of situation, and then when it will so Cre is expressing, for example, in macrophages, and over there is a LOXP site, both allele will having the LOXP site. So in those cells the targeted gene will be deleted. Okay. And once this gene is deleted, then you can achieve the knockout in that particular cell type. You can do it beautiful experiment using this kind of mice. Okay. So this is the concept for creation of conditional knockout mice. Eventually you will have this kind of thing, and then you will cross with Cre mice, and then you can create the conditional knockout mice. So this is a very complicated system, and I think the biologist, not only immunologist, they should know these techniques, and it is very useful not only for studying immunology, for any biological sciences using animal model, particularly the mice model. Okay. So, with this I will stop this transgenic mice. I have explained you the conventional knockout mice, how to create the conditional knockout mice, what is the uses of those conventional knockout mice and conditional knockout mice, and it is very important biological reagent in order to prove any phenomena or concept under physiological condition. Okay. Thank you very much.